Thank you.
Okay, so we're going to now do session 6, which is going to focus on the next part of the pathway, which is the ganglion cells. Let's see. So let me just click here. So again, I go through this with you every time. The whole point of doing this is to be able to see the the OCT, be able to list all the layers and effectively understand what's happening. So hopefully, we go through everything in such detail that it becomes pretty easy to know what exactly is going on here. Again, this is where we're looking. This is our OCT. Just remember, again, the retinal circuitry is complicated. Don't get nervous if you don't understand everything. Every time you're confused, try to slowly break down everything into little pieces, and then we just do it from different angles, which helps. We do that brief review. Last time we did bipolar cells. This is that schematic that I showed you before of the primate retina. So just remember, this isn't the human retina per se, but it is close enough. This is the primate retina. And so I told you that there were like 11 types-ish. So remember which direction the light is going. This is not done by MDs. This is done by PhDs. So the photoreceptors are on top and the ganglion cells or the nerve fiber layers on the bottom. So we go from outer to inner. And you see here that, remember, bipolar cells are classified in many ways, one of which is this off-center and on-center manner. So you have the off-center and on-center bipolar cells. And how do we know that now? We know structurally their axon terminals tell us that. If they're in the distal IPL, then you know it's the on-center type of bipolar cell. If they're in the proximal IPL, then they're in the off-center type of off-center bipolar cell. And then we also know that if we focus on their dendritic terminals, we get a different type of classification. These are the ones that connect to the photoreceptors. So if I said that, there's something that's flat, then you know that that's going to go onto the surface of that cone. Usually it's a cone, but that photoreceptor. And so we see one here, which is a flat bipolar cell. The F in that cell is for flat. But then we know that there's another type of cell when you look at the dendritic terminal in which they're invaginating. So we say, well, that goes within, that actually strikes within to that synaptic ribbon, if you remember in your book that diagram. And so this is the invaginating bipolar cell, of course. And then, of course, there's another way, if you look at the dendritic terminals, you say, okay, well, there's the midget bipolar cell. And the midget bipolar cell is just one single cone because midgets are monogamous in this terminology. And so you look here again, there's that flat, of course, but it's the midget bipolar cell. It's a small bipolar cell. And the same thing here, the invaginating one that we showed was a midget bipolar cell. That's what the MB stands for. And so you have that midget kind of smallish bipolar cell, one cone. But then you, of course, also have diffuse bipolar cells where you have a bunch of cones, and sometimes you can have some rods, in very rare cases, but it's a bunch of cones. And you can see all these ones with the D in the name are diffuse bipolar cells, diffuse bipolar cell 1, type 2, type 3, all the way up to type 6 in this case. And then, of course, you have your unique bipolar cells. And the unique bipolar cells, the most famous one is the rod bipolar cell that we discussed. And the rod bipolar cell is shown here for RB. And those are multiple rods. They're also, if you remember, despite the fact that there's multiple kind of diffuse connections, they invaginate into each one of those rods, if you remember correctly. And then they don't actually connect to a ganglion cell. They're that special type of rod bipolar cell or type of bipolar cell in which they use amacrine help to kind of use the corresponding next door cone on center bipolar cell that's sitting next to them. Do you remember all this?
Absolutely.
Okay. And then, of course, we have the other unique bipolar cells, one of which is that S cone. There's a special S cone bipolar cell, or we call it the blue cone bipolar cell. It's a diffuse on, and there's the BB where it's a diffuse on. And then you just remember before, I also mentioned that there was this like midget S cone that they thought was like a separate one. But in reality, we just think that that's just part of the flat midget bipolar cell kind of grouping. So we don't like specify that that's the S cone one, but just know that sometimes you see that. But remember, for this one, this is a diffuse because you can look at its dendritic tree. It connects to tons of them, and it's on because it's distal IPL axonal termination spot. And then, of course, there's that giant bistratified weird on-off kind of weird one where its axon terminals are in the both off and the on layers. So these are your bipolar cells. You remember all of those. So we kind of made a brief run through. Now we're going to do this again, which this is to remember the schema and what we're really doing here, how the actual retina works. So if I make it daytime and shoot a photon at the central. Now we know the difference between central and surrounding. The central photoreceptor, what happens? Is it active or inactive?
When it's light, it's inactive.
Correct, which means that glutamate is or is not secreted.
Is not secreted.
Is not secreted. Then which of the bipolar cells are activated? The on center or the off center?
The on center.
You got it. And then if I introduce these horizontal cells, just remember, are the horizontal cells in this state active or inactive?
I believe they're inactive.
Correct, because they're at the same party as the central photoreceptor. That's their guy. And then so he's inactive, these are inactive. What happens then to the surrounding photoreceptors?
They get activated.
Correct, they get activated. And then the bipolar on cell is active, the on center cell. So which of the ganglion cells are activated, the on or the off?
It is the. I'm not sure. Yeah.
Yeah. Just remember, if it's on, then this is where it gets somewhat easy, in my opinion. If it's on center, then the one it's going to activate is going to be on center as well.
Ganglion on cell.
Yeah,
right.
so there's no discordance with the ganglion cells and the bipolar cells, thank God, right? But they just kind of keep going up the chain. Thankfully, there's no weirdness there. And then, of course, we have our amacrine cells that are helping. And then those guys send off to the lateral geniculate nucleus. And then if we drop everything, if we like take off that schema, if I tell you the surrounding photoreceptor, and surrounding photoreceptor, those three boxes, where are their nuclei located?
These are on the internuclear layer.
The surrounding photoreceptors down here, which were these guys located?
Not those guys are in the nucleus is in the outer nuclear layer.
You got it, outer nuclear layer. So outer nuclear layer. And then their connections are in the outer plexiform layer, you remember? So I'm kind of showing it in this schema. So now if we drop down these guys, remember, where do all the masses live?
The masses are in the inner nuclear layer.
Right, so that's the internuclear layer, all these guys. And then where do they connect to the ganglion cells? And where do they make it? Their inner plexiform layer. And then those guys, the ganglion cells, where do they live? In which layer?
A ganglion cell layer.
Right, and then they send their axons through what layer to the LGN?
The nerve fiber layer.
Exactly. So there you go. So this is like, we just keep repeating this. I mean, and it's just going to keep being really, really like kind of just at some point, you've basically gotten most of it and you're just going to keep hitting it. Now I'm going to show you a cell because I always say these, but let's go through it together. What is this cell?
So that is a bipolar cell.
Think of where it starts.
This is starting on the Brooks. Oh, so that's the photoreceptor.
You got it. And then what's this cell?
That's the bipolar.
You got it. And what's this cell now?
Horizontal cell.
You got it. And that cell?
And this is the ganglion cell.
And this cell?
And the amacrine cell.
Good. And this cell?
Is the Mueller cell.
Good. And then these two cells?
That's RP Brooks complex.
Exactly. You got it. So photoreceptor, bipolar cell, horizontal cell, ganglion cell, amacrine cell, Mueller cell, RPE. You got it. Okay. So we're going to focus on the ganglion cell. Okay. So we are now on to, I think I said this is Dennis Rodman. So we're on the final of the big three, although I don't know why it goes that direction because Dennis Rodman doesn't score. So it's just like he rebounds and throws it back to the other guy. So anyway, but think of it that he does dunk the ball here. So ganglion cells, there are 1 million ganglion cells. There are 1 million ganglion cells is what I want you to just remember. So 0.7 to 1.5, but 1 million ganglion cells. They transmit from one neuron, bipolar cells, because that's where they're starting, to another group of cells. This is the main pathway, and I didn't talk to you about who Dennis Rodman passes to, but he passes to someone else. There's another person. When you score the ball, I guess that's like the referee or the other team, or I don't know what you want to say. But there's another person that he's handing the baton off to, and those are the LGN cells. There are 5 classes of ganglion cells, but there are also on and off forms that are independent of those classes. As we remember, the same way with bipolar cells, you can have a type designation that is separate from this on-off designation. And so you can kind of mix and match that. Bipolar on cells activate the on forms. Bipolar off cells activate the off forms. So we go, here's darkness. Darkness means glutamate is secreted. Glutamate being secreted means the bipolar off cell is active. That means that the ganglion off cell is active. The reverse is true when light is shining. When light is shining, a photon is shot, it hits the photoreceptor. Photoreceptor is inactivated or hyperpolarized, whatever. the glutamate stops secreting at the synapse, thus the bipolar on cell is activated, thus the ganglion on cell is activated. Does this make sense?
Yes.
Good. So we see this now. Remember, it's not just 5 classes, though. There are 22 actual types of ganglion cells, of which there are 5 cells. So 5 types, 5 classes. So, but there are 22 types, and those types keep growing as we discover them. Here is a primate, or maybe this is mammalian, so it might just be cat. I'm not sure. But the point I'm trying to say is that look how different these ganglion cells are. Each one of these could be sorted into different classes, 5 different classes, but each one of these looks so manifestly different. And that's just because the ganglion cells really, really are taking in a lot of information. So I'm showing you, here's one where it's like totally, different, right? And then here's one where you have it looking like this, and then one that looks like this. These are very different looking ganglion cells. So they're not just, again, so not to, just like the bipolar cells, they're not simply on and off. Like bipolar cells, there's this on-center characteristic and off-center characteristic, but they're not just that. They have this extra thing, which is not just on-center anymore. It's on-center off-surround and off-center on-surround. This is confusing. So we'll talk about it. And then there's this third on-off ganglion cell, which should kind of light something up in your head. Like, wait a minute, you told me there was this bipolar bistratified on-off. So it makes kind of sense that there's someone that he may hand off to that's his like analogy when he's handing off the information. So in the same setting, just remember there's this third one that we'll get into. But if we talk about this on-center off-surround, this is something that we kind of have to understand. So first of all, let's think about what a receptive field is. A ganglion cell is where the bulk of our processing is done. That's totally just understandable. It just takes into account multiple photoreceptor, bipolar cell, and amacrine cell inputs. So it is the clearinghouse of the entire retina. Here's a view of the convergence. You can take a look and see, this is not human, but just look, you can have a situation where you have 75,000, let's say rods, or 21,000 rods in one case, and then you have 5,000 rod bipolars, 250 amacrine cells that deal with those rod bipolars. And then you get this all coming down to one ganglion cell. So you can see, you can have anywhere between 21,000 and 75,000 rods, and these are all coming down into one ganglion cell, which shows you just how important those ganglion cells are. So, if you think about it, then the ganglion cell has a receptive field that is larger than any individual photoreceptor, and each one of these fields can vary depending on what that ganglion cell is supposed to do. Now remember, receptive field is talking in this case about the visual field that ganglion cell basically corresponds to. Does that make sense?
Yeah. Yeah.
Okay? So like if one photoreceptor is like, slightly to the left, and one photoreceptor is slightly to the right of center, and one photoreceptor is in the center, those 3 photoreceptors, if they send their information up and those just, kind of coalesce on one ganglion cell, that ganglion cell's receptive field is going to be all of what those photoreceptors are spanning in terms of the visual field. Does that make sense? Okay, so just to make sure, because sometimes when we get forward in the speech, or in what we do here, it gets confusing because you're now looking at, well, where are the ganglion cells processes in the retina? What is the field of those ganglion cells? Like, which cells are they touching? That is not a receptive field. Does that make sense?
Got it.
Because we're talking about the visual field that this corresponds to, not the pathological space of the axon spreading. Okay. All right. So, one of the reasons, I was just saying these things help with lateral inhibition, and it also takes into account horizontal cells. I throw that in there. Not exactly sure why I put that right there, but just remember that lateral inhibitions are for also horizontal cells are classically the lateral inhibition cell. So we're going to go through now, a on-center, off-surround example of a ganglion cell. The left side is basically the light I'm shining, okay? So right now I'm not shining anything. And it's gonna be this like annulus kind of situation where I'm gonna shine a light on the center, and then I'm gonna shine a light on the annulus, and then I'm gonna turn off the annulus kind of thing. And so the annulus is gonna be the surround, if that makes sense, and the center is going to be the center, right? So that's what it's gonna be. So let's try the first one, okay? So I'm just gonna do one of these. And this right now, is a basal level of firing because there's nothing being shown. This ganglion cell is like in the dark state. There's nothing, there's no light. It's just sitting there, okay? So here's
Okay.
your basal level of firing. Now notice something. If I were to put a probe in the ganglion cell, it's still firing, okay? It has this like baseline, almost like, you know, like you said, basal level of firing, like a ganglion cell is just telling you it's alive right now. Okay, so if you were to have all of these ganglions, if these ganglion cells just bathed in darkness, you would see a firing pattern that's just telling you, hey, I'm here, you know, I'm not dead, I'm alive,
Yep.
I'm gonna keep shooting. Okay, so that's how it is. We'll just show you again, you can hear it, you know, there's your firing, there's your firing. It's just kind of, there's no rhyme or reason. It's just sporadic enough that you know they're alive. Okay, so now we're gonna turn on the center here, right? So I've shown light on the center, and he's a ganglion on center, which means he likes, quote unquote, you know, I don't like the term, but it's easy to use the nomenclature here. He likes when the center is on and the surround is off. This is perfect. This is like if you were a baseball player, this is like he's going to hit a home run, right? He's so happy, right? This is where you would expect something major and different to happen, right? And so here's what it looks like. Light goes on and you see this massive firing. And then you turn the center off and then it goes back to basal. Does that make sense?
Got it.
You get a high burst, okay? The ganglion cells.
Sorry, Dr. Deepak, that firing on a group of photoreceptors that corresponds to the ganglion cell on center. Yep.
Correct. Okay. You got it. And a group of photoreceptors that correspond to the center part of the receptive field of this ganglion cell. Make sense?
Yep.
Okay. So this makes sense. And I just like doing it again, feeling it again, basal, on, major, because this is the way, when we say on and off in this case, when we're showing on and off, we're not just saying on and off. This is just representing this kind of a fire. Okay. Now we'll see a kind of a weirder one, right? Now I've turned everything on. So I've literally, when I say on in the firing pattern on the right of your screen, I don't just mean on center, I mean on center and on surround. So in this case, it's like the ganglion cell is telling you, well, you know, that's great, I like this, but it's not as good as the last one because you can see it in my name. When do I hit home runs? I hit home runs when it's on center and off surround. But this is on center, on surround. So you'd expect something to fire, but maybe not as good as what you saw before. Does that make sense?
Yep.
Okay, so here, let's watch what happens. So we play, and then all of a sudden it's like, okay, like that's definitely more, right? It's definitely more than your baseline, but it's not like that much more. You get what I'm saying?
Yeah, totally.
Okay, that means that light was being shown both in the center of the receptive field, the photoreceptors that correspond to the center of the receptive field, as well as the ones on the surround that are being lit up too. And by the way, this is telling us something different, despite the fact we're getting more information, a little bit more granular information from this ganglion cell than simply on off, right? You're getting spatial recognition of the light, right? You can tell in this case, if I just showed you the firing pattern and you know what type of ganglion cell this was, you'd say, oh, that looks like it's not basal, that's a lesson test burst. Maybe this is the lights on in the center and on the surround as well. And then if we do this, this is like everything this ganglion cell hates. If you can imagine, right? So light is actually quote unquote on, but it's in every way that this ganglion cell dislikes. Okay, so what would you expect? You'd expect literal, here's basal level, inhibition.
Got it. Yep.
And then here's this weird thing, how when you shut this back off, meaning you turn off this annulus and you go back to that kind of dark state, see how you get like a burst of firing?
Yeah.
It's the ganglion cells telling you, because it has this time-sensitive like dynamic ability, it says, hey, not only did I hate that, you inhibited me, I was suppressed, but when you unsuppressed me, I want to tell everyone I've been unsuppressed. Look how great I look. And then it goes back to basal. So there's that piece of information that it's feeding forward as well. Does that make sense?
Yeah.
So, and you don't have to ask me why this happens. Just know that it does. Meaning the ganglion cell has the ability to do this. It's that sophisticated, okay? And again, I always tell you, these actually were figured out via very classic experiments, right? So that's why I'm presenting them to you, but it makes sense because the ganglion cell is taking into account a bunch of more cells. So it should have a more kind of sophisticated firing mechanism than a photoreceptor, which just kind of takes in light and then, you know, secretes glutamate or doesn't secrete glutamate.
100%. Yeah, that makes sense.
Okay, so now, and thankfully the canonical experiment was on center, off surround, and not the opposite, which would be like harder to think about. So this makes a little bit more sense. Okay, so again, just to know, it would be reversed in the reverse situation. And then remember this, sublamina, what we're looking at, here's your cone, here's your bipolar cell off, bipolar cell on, sublamina A, sublamina B, which direction is everything going, right? It's going from bottom to top because this is being drawn by an MD. So if you do this, there's a ganglion cell correlate, right? So here we're going to do this. We're going to actually make sublamina A and B a little bit bigger. And then we're going to split up these guys almost arbitrarily into something called S1, S2 for sublamina A, S3, 4, and 5. Now remember, which direction is the electricity being propagated? Electricity, the signal is going this way, right? It started down here at the photoreceptor, and then they're handing off the baton this way. So the numbers are being counted in that way, just like sublamina A and sublamina B when you're talking about it relative to the bipolar cells. In the same way, when you're talking about it relative to the ganglion cells, you can still say sublamina A and sublamina B, but I'm just saying at the end of the day, your ganglion cell layer is able to have that same correlate as well. So if we show this, here's an off, right? And that's where it would theoretically have its dendritic process, right? Because that's where it's getting its information from whomever, usually the bipolar cell. And if it's an off, it needs to hand off or have its hand or hold the hand of an off bipolar cell. So it makes sense that it would sit in sublamina A somewhere, and then on cells are sublamina B, and I'm throwing, you know, S4 for this on and S1 for this off, but, you know, and just to show you, that's how this would draw, I would draw this maybe. And, you know, it doesn't have to be here. This guy could be in S2 as well, if there was a, you know, a bipolar cell that connected to it in S2. Does this make sense?
Yeah, totally.
Okay, so there's that ganglion cells dendrite. If it's an off, it has to hold hands because you already told me that it gets activated by the same like valence, so to speak, or the same like the off on bipolar cell. If it's on, then it's going to activate an on, right? And so if it's going to activate an on, it needs to hold hands, like somewhere that synapse has to be. Well, it better be in the same spot, otherwise how does it activate it, right? So it kind of makes sense in this case, right? On would be both, the connection would be in sublamina B, or in this case S4, S3, 4, 5, and off would be in sublamina A, S1 or 2. Make sense?
Yeah, I think so.
Okay, so I'm going to show again, this you can be connected anywhere here. So here, just the correlate again, bipolar on off, translates to ganglion on off. Sublamina A is S1 or 2. Sublamina B is S3, 4, or 5. That's it, right? I mean, I'm just showing you, that's kind of how it looks. Okay, so if we look over here, I'm showing it to you from a different angle. Now tell me, did an MD write this or did a PhD write this?
It's going the opposite way, where the signal is at the top going down. So it was a non-MD PhD.
Right. Well, it was a non-MD. It was a
PhD. Yeah, it was a PhD. It was a non-MD.
Exactly. It might be an MD PhD. I don't know. Yes. They kind of go both ways, but an MD would not have written it this way. But there's, you know, these are very famous people, and they drew this kind of for us. You can see up here, right? First of all, it's upside down, so to speak. This is the direction of the signal propagation from photoreceptors onwards. And then you can say, here's your off ganglion cells, right? Because this is the ganglion cells down here. And so where are they synapsing? If you follow them in, they're synapsing in S1 or S2, which is also called sublamina A when you're talking about bipolars. And then if we do the same thing and we say, okay, here are our cones again, let's look at the ganglion ons. Well, I know they're on because they better be synapsing in S3, 4, or 5, which also corresponds to sublamina B. Make sense?
Mm-hmm. Yep.
Good. So here are the 22 primate, supposedly, or cat or mammalian, I'll say, ganglion cells. Now, one of the things that you have to remember is they divided up. First of all, again, you tell me, look at, just kind of figure out what you're looking at here. You're looking at and I'm going to let you kind of look at this. Who drew this?
Right.
Is it an MD or a PhD?
Also a PhD.
Correct, right? Because the GCL is down here, down, and the INL, which is where the bipolar cells live, they're way up here. And here's your IPL, which is being split up. Now, mind you, look at this. This is 0 to 20, 40, 60, 80, 100, just assuming, and I believe this is true, that they are equidistant from each other. Now you understand why it's S1, S2, S3, S4, S5. Makes sense? And then now S1 and S2, this is the proximal, as we would call it, closest to the outer, like sort of near the photoreceptor. This is the IPL, and you sit here and you say, oh well, I guess that's where off is. I guess the off is actually smaller than the on, right? I mean, it kind of has less real estate, right? I don't know why the body did that, but it has less real estate. So I'm going to just did this whole thing with you just now, but now it's going to make sense. I see 5 divisions. I'm doing the same thing over here. Well, this one probably is S1, and this one is probably S5. Okay, and then that's S2, and then that's where your dividing line. And so thus that's going to be sublamina A, and that's going to be sublamina B. We just did this on a graph that didn't write any of that down. Make sense?
Yeah.
So now we're going to play a game here, because now I'm literally grabbing this from, I'm forgetting which PhD textbook, but we are legitimately able to make inferences here that other people may not be able to make. But let's go ahead and look at this one. I am just drawing this here based on what you know. Can you tell me, is this an on or an off?
So that one goes through all the way to layer 5, so it's an on cell.
You got it. That's an on cell. How about this guy?
Oh, sorry, that was layer 4. This is also layer 4, so it's also an on.
Yep.
Oh, no, sorry, we're going.
You're right.
Yeah, sorry, we're going. Yep, perfect. Okay.
You're right. You're right. You don't have to second guess yourself, Nathan. This is like, you're like, wait a minute. It's like magic. I mean, I always say, like, you get to a point where you're like, wait, why are we actually making these inferences? Like, no, they didn't make these inferences here, but we are. And by the way, it's true. So what about this one, on or off?
It's an off cell in S2.
Yeah, that's an S2. It's off, exactly. And then what about these two guys? Are they off or on?
They're off as well, S1,
Correct.
S2.
S1, you got it, off. And then I'm just gonna just have fun here. What is this one?
This is a combined cell.
Like a bistratified off on, right?
Yeah, yes.
Yeah, okay, good. We're like, grr, okay, that exists. But now like I always say it's just astonishing. You legitimately are looking at a sheet that you just learned about 5 seconds ago. And now you're like, I can make 50 million type of inferences on this, very specific things about the functionality of these particular things. I may not know necessarily what on is, but you already know how an on center, it's not on, it's on center off surround. I told you that. That's what an on is for ganglion cell. I showed you how it fires. So any one of those things that you told me at the beginning, oh, that's an on, you can also already tell me what its physiologic burst pattern is just by looking at this. Isn't that crazy?
That's great.
I think that is very powerful when it comes to the retina, that you can literally determine specific physiological, electrical input or specific physiology based on where their location is in the interplexiform layer, literally. And that's really cool. So, and then this one's bistratified, we said. So I said, by the way, it doesn't mean that each one of these are 50. Like I said, here's an existence of a cell. It's kind of sitting at the same, it's on the same amount. It looks like there's G1 through G22. It doesn't mean that there's bistratified cells are 1/22nd of the population, okay? These are very rare, but they're just showing you that they exist, okay?
Got it.
Right, and just remember, the vast majority of these ganglion cells are either on or off. They're not on-off, okay?
They're not bistratified.
Yeah, and remember, on-off does not mean on center off surround. That's not what on-off means. On-off means something weird. On means on center off surround. That's what it means. I'm just not saying the rest of that sentence. Does that make sense?
On means on center off surround. Okay, yeah.
Right. It's like a shorthand for that. When we say off.
And off means off or on surround. Yeah.
Right. And then off on, this weird one is weird. We just know that it's both of the qualities of off and on that we've just discussed.
Got it.
Okay. And you don't have to care about off on just yet, because it doesn't really make sense in the way of the way I described it, but it's just keep it that it exists. Okay. All right. Now we're going to go through classes. This one is I don't know how to say that. This is just one of those times where you just have to be like, yeah, they named it like this and everything got named this way. And I don't know, I'm sorry. It's what I always say to you guys, but we're going to run through it anyway. Okay. So here are the classes. There are 5 different classes. I do my best to color code them and do all this stuff. Okay. I always say they're unfortunately named because of how they act and what they look like. So here's the first. Okay. It's called a P-type parvocellular pathway. Now, the type and the pathway always correspond. The pathway is what helps you, but they're midget cells. They're midget ganglion cells. A midget ganglion cell is not a midget bipolar cell. Okay, I don't know how else to put this. They're not. That's just the way they looked.
Does this have one connection?
Well, I wouldn't even go there. Midget bipolar cells, I should have said midgets are monogamous when they're bipolar cells. Midget ganglion cells are not monogamous, but they have a very specific kind of functionality for sure. But I'm just telling you, they're midgets because they look small. And the same was true of your bipolar cell. But with your bipolar cell, you had an extra thing you could infer, which was one code. Okay. So this, we're talking again about ganglion cells, right? A P-type parvocellular pathway equals midget ganglion cell. Okay. I also made this purple because it's P-type. Okay. So purple. Okay. P-type. Now, here's where everything just flips. Here you go. Thank you, nomenclature people. The M-type magnocellular pathway is also called a parasol ganglion cell because of the way it looks. Parasol means umbrella. So you've got a P-type cell that is specific to the parvocellular pathway, which again, we'll talk about later as to why it's called that. And then you've got this M-type cell, that's the magnocellular pathway, that's specific to a pathway that we'll talk about later. But what they actually are when you call them their pathologic designation is midget cell and parasol cell, which are the opposite letters.
Amazing.
I didn't, yes, I'm sorry, okay? There you go. This is just how it is, okay? Then you've got this thing called the K-type coniocellular pathway. These are the 3 main pathways. I've put them up on the screen. They're the only 3 that you really know, and you have to know them, okay? Because they connect to everything in the brain. They help you with your neuro-ophthalmology. They'll help you with glaucoma. They're going to help you in retina. They're going to help you just understand the visual pathway. But this guy has what's called the bistratified cell. Sound familiar? This guy's ganglion cell is that bistratified weirdo.
Yeah, got it.
Right? But remember, we are talking about ganglion cells. We are not talking about bipolar cells, albeit the bistratified bipolar cell is connecting to this as well most of the time. But I'm just trying to explain, this is a ganglion cell. The ganglion cell is bistratified. It's the K-type coniocellular pathway, okay? So you've got these 3 pathways. They are like this. You're going to be hammered over the head about this. It is what it is, okay? So that's that. Now you've got this weird one called the photosensitive ganglion cell. This is the one major exception to the rule that I said before, which was the only cell that actually takes in photons is the photoreceptor. Oh, except for this guy too. Okay, it's you got your photoreceptor doing 99.99% of all the photon acceptances, and yeah, the photoreceptor is there. That's why they're called receiver of photons. They're the photoreceptors, and then no one ever receives photons. Oh wait, except for this photosensitive ganglion cell, which is weird. Okay, you just have to remember they exist. And then the final one are these ganglion cells that project to the superior colliculus in the midbrain. These are your 5 types. Okay, they are your 5 clearinghouses. Okay, nothing gets past them. Okay, there's literally no way to get to the brain unless you go through these ganglion cells. Okay, so everything gets to these. Okay, you got it? Now, mind you, I very much presume in about 10 years, someone's going to be like, well, Dr. D, they discovered a 6th type. I'll be like, okay, thank you. But as it stands, you got to go through these. Okay, we're going to go through these. Okay, so yeah, and I don't want to talk about this confusing historical non-human correlate, but it exists. And oh, by the way, it's equally as confusing, and I hate it because if you ever read any of the texts, you have to figure out which one is which. But we're not going to talk about it just yet. Okay, so I'm showing these 2, and I said P goes with M and M goes with P. It's so bad, but it exists. I don't know what to say. Let's go through the first one. Here's your These are 80% of all ganglion cells, okay? 80%. They're your major ones, okay? They're midget ganglion cells. They look small. They have small dendritic trees and cell bodies. That's why they're called midgets. When you look at them on a pathological specimen, you'll say that's a small one, okay? They project to the parvocellular cells in the LGN. That's why they're called P-type. That's why they're called the parvocellular pathway. It's because of those cells in the LGN. That's, again, why they're called P-type. They are responsible for color vision. So the vast majority of our apparatus is cleared through these ganglion cells who deal with color vision, but they're not fast. They're slow because it's color vision. Their job is to be detail oriented. There are very relatively few rods and actually small amounts of cones for every single midget. So when you asked me, oh, there's only one dendritic connection to these midget cells the same way the midget bipolar, that's not true. But if you wanted to have a correlate of a monogamous ganglion cell, it would be the midget ganglion cell. Make sense?
Because there's few rods and cones as input.
Yeah, and also they're small. So if they're small, they're not going to have a lot of connections anyway. So it makes sense that a midget, cell would have fewer connections. So in this case, it's not monogamous, but it's as close to monogamous as you can get. Okay. So this is where we make that nice correlation. That doesn't mean that only midget bipolar cells connect to midget ganglion cells. That is not what I'm saying. I'm just saying that they're very similar in terms of what they do and who they connect to. That makes sense. Yeah?
Yeah, totally.
So these guys tend to be, and here's my line, that's why I throw they're dealing with the fovea. These ganglion cells are dealing with the input from the fovea a lot of times. Sometimes, and they're much smaller than their counterparts. And yes, sometimes they receive input from one bipolar cell, but not all the time. It's not like a midget bipolar cell where it is true that one cone is what it touches. Okay, it is sometimes true that it touches one midget bipolar cell, but not always. Make sense?
Yeah, totally.
Okay, this is where we're looking. Here is a primate retina, and we're looking at these cells, and this is how they figured these cells out. But here's your midget 8 millimeters away from the fovea, where they're a little bigger. But notice, compared to the guy next to him, he's pretty small, right? Makes sense? He's pretty small, right?
Yep. Yep.
Sorry. Yeah, he's this big, and then you've got this pretty large guy, right? And we'll talk about him next. And then even if you go here, he's pretty small. Now notice again, what is so confusing? Midget P. Okay, midget P, and then the next guy we're gonna talk about is M parasol. Do you see the confusion already that's getting in your head? This happened to me many times, but just know that it's normal to feel this way because this is the way it's named. So P goes with M and M goes with P. If you ever have a PP situation, you're messing yourself up a little bit. Okay, so the way I try to explain it or try to advise is when you're going through this, pick one way of talking about it. The way I tend to pick it kind of fluctuates because I've been doing this for a while. But what I tell you guys, go with this one, P-type. Because if you go with P-type, then you already know what the parvo cell, it's the pathway, and then you remember the pathway and what it's responsible for. It doesn't change. The pathway is responsible for color vision and slow conduction. You can follow that pathway all the way out. But just know they're going to try to throw you some loops sometimes by throwing these midget words in. Just know midget equals P. Okay.
Yep.
Okay, here we go. Next one, the flip, or his counterpart, M-type parasol cell. M-type parasol cell. Again, this is only about 10%. Let's say 12, so that way it's second. Okay, because the other one's gonna be 10, and then it's like, wait, which one's second? This is the second most common as we think of now, I believe. Someone may dispute that later, but I believe this is second. Okay. These are called parasol ganglion cells. They have large umbrella-like dendritic trees. That's why they're called parasol ganglion cells. But they project to the magnocellular cells in the LGN. Now, we'll explain why these are called magnocellular, parvocellular, and coniocellular later, because that's when we get to that organ. But right now, the magnocellular cells, just know that these guys are handing off to the magnocellular cells. So that's why we call them M-type. Okay? It's the magnocellular pathway, which is responsible for motion. Motion. right? Magnocellular pathway, motion and low contrast. If it's motion and low contrast, it's fast conduction, and it makes sense that it would be rods. Does that make sense?
Fast conduction. Yeah, absolutely.
Because rods, remember I told you about when we talked about rod bipolar cells, the rods are responsible for kind of night vision. Well, when you get vision in the night, you need to see that thing really fast. And you don't necessarily care whether it's what color it is, but you definitely want to know it quickly, and you want to know if that thing is moving towards you, right? And so I always think of this as like night vision. This makes sense. The magnocellular pathway is kicking into high motion, right? I want to see something. I don't care what color it is. I care that it's, moving. So thus, this is the pathway that does that. The magnocellular pathway, the M-type ganglion cell, motion, fast conductance. I want to see something. If it's moving fast, you better be fast. And guess what? Those are rods.
Got it. Great.
Right? And it's got to have a ton of those. So it kind of makes sense that this is it's a parasol cell. It's going to connect to a ton of things because it really needs to see if something flew across your visual field. It's probably going to have a huge receptive field, right? And it's probably going to be like, it's a big receptive field. I'm going to figure out whether something went from left to right really fast, but I don't necessarily need to know the color. So I'm going to be like a diffuse bipolar. So I get a lot of connections in the retina connected to tons of rods and maybe some cones. I'm a ganglion cell. I don't have the same rules as the bipolar cells. But I'm going to go ahead and I'm going to connect to all these things. And if something really quickly happens, it's really fast, I'm going to send that really quickly through the M pathway, the magnocellular pathway. Make sense?
Yep.
And then if you look, here's your parasol cell. I mean, look how much bigger this is compared to. Now again, parasol cell, M, right?
Yeah.
I always say, think of it as an M-type cell, but then remember parasol.
Yeah. Okay? I like that.
Right. Now we're going to the next one. Case types bistratified, the weird one. 10% of all ganglion cells, let's say it's 8, just so you know it's number 3 on the list in terms of frequency, okay? I'm not so sure they've made a distinction just yet, but just 10%, okay? So just, it's number 3, but it's very close in terms of frequency.
Sure.
Bistratified, what does that mean? You know what the stratifications are now. It's in the on and the off. That's why they're called bistratified. Now you know, like when you read in your book, if you have, they'll talk about the bistratified. They'll talk about these K types. You'll be like, okay, I've just got to memorize it. Now you know exactly what they mean. You know exactly what they mean. You'll be able to tell exactly where this thing is if you were to see a layout of the IPL. But here's the weird thing. This guy deals with something odd. It's always on to a blue cone that's sending its stuff and always off to some red-green cone. So he's kind of like, he's kind of like got tendrils in both. So and then he projects to the coniocellular cells in the LGN. So he's called this is the K type. So remember, this is responsible for some color vision. We think blue-yellow, okay? We think blue-yellow kind of vision, and some kind of integration, kind of somatosensory, sort of proprioception, close your eyes, you know where you are, kind of weird stuff, right? It's meant for that. It's a special guy. He doesn't necessarily do, but he's always on to the blue and off to the green. This is the idea. His on dendrite, dendrite is connecting to a bipolar cell, like what I told you, those S-cone diffuse bipolar cells, that itself is connecting to a bunch of S-cones, which is a blue cone, and sending the blue cone and awning that, and then offing the other ones, because blue cones were kind of later in our development as evolutionarily. So these guys are a little bit more kind of newer and weirder. Does this make sense?
Yeah, I think so.
Good. Yeah, I mean, remember, you don't have to understand why we made our body this way. It just is. You know what I'm saying? Like, it just is, right? So, thus, this is the exception. You know that there's no surround here in a weird way, right? Because you knew that if I tell you you are on, then you know that the surround is off, right? You have something special with the surround. And if I told you you were an off, then you had an on for the surround. You already knew that. That's just the way the nomenclature was. But if you're off on, it's like, wait, is your surround off or on? Well, you have no surround.
Yep.
So it makes sense that this would have no surround. There's no surround that's changing the nature of the central vision. So thus, it has a huge receptive field, and it doesn't have any surround that's making some kind of inversion of that. Does that make sense? Yeah,
totally.
Because now you know how physiologically a ganglion cell works. When I showed you the bursts, you understood, oh yeah, the off surround kind of has to be off and the center has to be on for me to have the highest burst. But if you turn on the surround and you keep the on center, it's not going to be as good of a burst. It's kind of a less intense burst. Well, this guy, he has no surround because he's off on. There was no surround. Nomenclature wise, that kind of hints at it. And so now you're like, well, it's just anything that's in his field, he's going to just turn on and tell you it's happening. You get what I'm saying? So in the end, he has no surround and has a large receptive field. He has intermediate amount of rods and cones. So thus, the only reason why this is here is it says, if you ever ask, like, which of the ganglion cells has the most amounts of rods and, of rods and cones, it would be your M type, your magnocellular, right? Because it's most of the rods are going there. But what about the fewest? Well, that would probably be your P type or your midget ganglion cell, the one that deals with color vision. But then who's in between? Well, it's this guy. This is why I say that. That was in your book. Now, again, this is the correlate in the bipolar cells, this giant bistratified bipolar cell that but it's not always that guy that's connecting with this guy. This guy can also just connect to a S cone for the on and then a red-green cone situation for the off. Make sense?
Yeah, got it.
Okay. So here is another kind of schema that I got from a book. So you see here, they're showing you the different types of ganglion cells, and here's your bistratified. I'm just showing you, here's a bistratified type of ganglion cell. Okay. And then there's your midget, right? And there's your parasol. Showing the dendritic trees. Remember, this is the IPL, therefore it's dendrites when we're talking about the ganglion cells. That's the way I like to say it. So it makes sense which direction we're at. And bigger for parasol, smaller for midget. Okay. All right. Yeah. Now here's your IPGCs, or your intrinsically photosensitive ganglion cells. Okay, here's the weird one. This is the odd one, but the nice part about ganglion cells is that they start allowing you to understand functionality a little bit better. So these guys are weird, fine. They're super weird, fine. They are 1% of all ganglion cells, very, very small, okay? Very small. But they have their own pigment. It's called melanopsin, okay? Now, can I just mention the most famous opsin? When I do my phototransduction series of lectures, I talk about the most famous opsin. It is the most famous. There's something called a G protein coupled receptor in all of medicine. The classic form is an opsin, or the classic form of it is the one that rods use, called rhodopsin. So rhodopsin is the rod opsin, okay? Opsin just means, I'm going to be very basic here right now. Opsin just means the thing that allows you to respond to light. Okay. The thing. Okay. But then you have the colors, and the colors are, and we don't actually specify them, but the colors ones are your S, right? Your S opsin, which is your short wavelength, your M opsin, and your L opsin, right? Your medium wavelength and your long wavelength. Okay. That's what we define them. So on a test, they always ask, how many opsins do we have? And the answer that everyone says is 4, and they get it wrong, because guess what? These guys exist, and they have something called melanopsin. Totally different. It's a different opsin. Opsins, again, receive light and convert them into electrical energy. They are the types of cell apparatus within a cell that can do that. Okay, so we call these intrinsically photosensitive retinal ganglion cells, which makes sense because they're intrinsically photosensitive. Or another way they refer to them is MRGCs, which is melanopsin containing retinal ganglion cells. Make sense?
Yeah. Yep.
Okay. So if you see these two, you know what kind of RGCs, what kind of retinal ganglion cell they're talking about. They can respond to light in the absence of rods and cones. Now, why does this, and then where do they go? They actually project to places like the suprachiasmatic nucleus, the hypothalamus, the Edinger-Westphal complex. Notice what I didn't say. I did not say the LGN.
LGN, yeah.
Right? Now, this is where you can remember, and let me just go through this, is concerned with ambient conditions, right? So it manages circadian rhythms, melatonin suppression, pupil size. Here's my favorite thing. When you see a patient who has lost all of their, think of a patient, let's say, that has lost all of their photoreceptors. Do you know of a patient with a genetic disease that could have had problems with their photoreceptors? Have you ever seen one? Famous disease. It looks like pigmentary changes in the periphery of the retina. What is a famous disease?
RP?
RP, retinitis pigmentosa. And do you notice something? When these patients are LP and they've lost basically all of their photoreceptors. Their pupils still work, right?
Interesting. Yeah.
They don't
Yeah.
have
Yeah.
APDs.
Interesting.
Why?
Yeah.
Why don't they have APDs? It's because these guys exist. Does that
Huh.
make sense?
Yeah, that's cool.
Isn't it? So I always say this is the clinical correlate. Now you understand why are they, one argument could be, well, they didn't kill all of the photoreceptors. And by the way, that was the argument that we used to make. They didn't kill all of the photoreceptors. Genetically, the photoreceptors, there's like one tiny amount that we can't see that still exists. But then people were like, wait a minute, but these guys are the ones that exist too. And wait a minute, they're not actually going to the LGN, they're actually controlling the pupils. And they're controlling the ambient conditions. It's when you get such severe destruction of the entire retina that these guys finally go, is when you start losing your ability. You go NLP, you lose your pupillary response, that sort of thing. Okay, so these guys are very important, and the major thing is melanopsin. They project to weird places like the Edinger-Westphal, not weird, but places that are not the LGN, like the Edinger-Westphal complex, hypothalamus, and suprachiasmatic nucleus. Okay, let me do this and do this. Okay, and so an example of this guy is the giant retinal ganglion cell, which is more like Bigfoot. Okay, I'm not even sure we've ever found it. I think some guy found it like in 97, but it makes up like 0.2% of all RGCs that are these intrinsically photosensitive retinal ganglion cells. This is what I'm going to use as a manifestation of how these guys look. So when I show you this kind of retinal ganglion cell kind of schema here, this giant one, right, a giant one, right, tends to be these retinal, intrinsically photosensitive retinal ganglion cells, or you can also say melanopsin containing retinal ganglion cells. And you can imagine why they're so giant, right? Because they're trying to get light from every part. Their receptive fields must be humongous because all they're trying to do is manage your circadian rhythm. They're kind of like those baseline kind of retinal ganglion cells to keep the lights on, so to speak. Okay?
Got it.
All right. And then these other guys, they're very rare. I'm not sure we found them. I think we know that projections occur. They project to the superior colliculus, which is not the LGN again. So these last two classes do not project to the LGN. They are made about maintenance of saccades. So the answer in this case is this is the ganglion cell that projects the superior colliculus, which deals with saccades. So superior colliculus, saccades. There's an S and a C and an S and a C.
Love it.
Makes sense. It makes sense, right? Okay, so we're going to go through these classes on another way. Midget, small, but again, P-type, little, okay? Parasol, M-type, parasol, umbrella. So as compared to these two, these guys are the two main ones. This guy has the bigger dendritic tree. Bistratified, K-type, this is my schema here, kind of shows you the cartoons. Bistratified's got two stratifications, and you know what that means. Here's the IPL, okay? This is the inner plexiform layer that we're talking about here. Here's your IP-RGCs. I use the giant retinal ganglion cell as my example, and then you've got this other where we don't really know what they look like. So these are your 5 classes of ganglion cells, okay? And then I hate doing this, but so I don't know, but I'm just going to do it. Okay, there are other nomenclatures thrown around. Because of the way these things were discovered, you will hear people throw weird stuff out, and it's just going to make you kind of be like, oh God, like, you know, what does that mean? And it actually took me a while when I was like doing all this study by myself, trying to like figure out like what are the non-human correlates. I suppose now I do rabbit research. Maybe this is why I do it, but like, you know, and that's why I'm like interested in it. But you have to remember that like it wasn't humans first. Okay, and not all the retina and the visual pathway is very different from species to species. So I'm sorry for this, but I'm going to show you it. Here is your nice, here's like the primate, and here is your cat who, you know, for whatever reason was done first. So a cat retina is on the right here, and your human primate retina is on your left. And if you look, here's at our fovea. We call ours the fovea. We call theirs the area centralis. What they labeled is that your midget P-type ganglion cell is also called beta. Now, I'm going to tell you this right now. There's only one reason why I'm bringing this up. It has nothing to do with you understanding cat retinas, okay? There is one time where the payoff comes, okay? For real. And it is something in the brain. So that's why I'm bringing this up, and then you'll go, oh, okay. And hopefully then when you get tested on it, you're like, I totally understand now, okay, Dr. D? So this is why I bring it up. Midget P-types are also beta. Why are they beta? Because I'm almost sure this guy was discovered first because he's huge, okay? And that huge one is correlating to our parasol cell, which is the M-type. Makes sense? There's your M-type and there's that alpha, okay? So they call it midget, which is your P-type, which is called beta, and another group of people call it X. Your parasol is alpha, another group of people call it Y, and then your bistratified K-type is your gamma, another group of people call it W. I'm just saying they exist, but just remember these two, there's a payoff later. I'm being honest with you. There's a legit payoff later in how we label locations in the brain. So if you can remember, this is the first time you're getting these. I will hammer it home again and again, but this is the first time you're getting this nomenclature. Midget P-type, which is your parvocellular pathway, is the beta cell, because the first one that was discovered in the cat, I believe it was the cat, at least this is what it suggested to me when I was reading about this, in the cat, the first one, the bigger cell, of all the frequent ganglion cells they were going to come across, the biggest one, the one with the largest dendritic tree, is going to be the parasol, because that's how big it is. So they called that one alpha. It's the parasol. It's the M type. You can forget the Y, but that exists too. And then, of course, our bistratified guy became gamma because, he was the third one.
Sure.
All right. So just know that, okay? And people will also randomly use that nomenclature occasionally when they're discussing primates, which is actually not right. Okay, so just again, the most numerous one is the P-type, midget, parvocellular pathway, ganglion cell. Parvocellular pathway is slow conduction, color vision. Okay, if you remember that.
Yep.
That is not the alpha. Why? Because it was not big. It was not discovered first. It's the other one. It's the magnocellular pathway, slow, fast conductance, motion. That's the M-type parasol alpha ganglion cell. Make sense?
Yes.
Okay, I'm just repeating it over and over again. Here it is. Okay, this was in a book. I saw this. Okay, what the heck does this mean now? Right? 20 to 100 cones. That's a tip-off. Goes to 3 to 15 cone bipolars. Okay, which goes to 1 beta ganglion cell. Well, now we know this is a midget. And it totally makes sense now. Cones are for color, slow conduction, totally makes sense. A midget, probably a midget bipolar cell. In this case, they're probably not midgets because they're connecting to a bunch of them, but a midget is probably a diffuse one. But then he's connecting to a midget ganglion cell, which is P-type, or the parvocellular pathway. Makes sense?
Yeah.
If these were a bunch of rods, that would be different probably.
Yeah, both of those are midget P-type. Even the one on the left is a midget P-type.
Even the one on the left is a midget.
Yeah, right.
Exactly. Even the one on the left, you got it. Okay, so just going through it again. On versus off, which is on center, off surround, versus off center, on surround. And then you've got this on off that has no surround, okay? Has no surround, and that's your K-type bistratified coniocellular blue weird color proprioception somatosensory ganglion cell, okay? And then lateral inhibition is based off of receptive field. I call it surround suppression. You don't have to care about that. On is S345 or sublamina B. Off is S12 or sublamina A. Dendritic placement is what tells you that, because it's a ganglion cell dendrite. Placement in the IPL. Again, we're in the inner plexiform layer. Never forget, that's where we're hanging out right now, okay? It didn't change because we're now through the lecture, okay? 5 classes of cells, going through it again. Most numerous is the midget ganglion cell. We're talking about the red-green color, the parvocellular pathway. We also call it P-type. The second, tied for second, or I would argue the second most numerous, is the parasol ganglion cell, which is your M-type, which is motion, low contrast, magnocellular pathway. We're not talking about color vision here. We're talking about just low contrast, motion moving back and forth. Tied for second, or I put them third, is the bistratified ganglion cell, which is on off, no surround, very large receptive field, blue color, weird integration, blah, 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 like proprioception, kind of nebulous stuff, integration stuff, coniocellular pathway, K-type. Then these two last ones do not go to the LGN. You've got your intrinsically photosensitive retinal ganglion cells that have their own opsin called melanopsin. They don't need photoreceptors. They deal with things like your pupillary reflex, your circadian rhythm, ambient light responses. They go to the Edinger-Westphal complex. They go to your hypothalamus, at suprachiasmatic nucleus, the things that deal with those sorts of things. And then your others, which I call the others, they deal with saccades. Why? Because superior colliculus deals with saccades, SCSC. Okay, you got it?
Yep.
Okay. Okay, now we're just gonna play. Okay, we're looking at this exact graph again. Okay, nobody else would be doing this except you now, because you kind of sort of know. I'm gonna point to something. You're gonna now try your best to tell me whether it's on, whether it's off, or on off.
Yep.
And then whether it's a midget or a however you choose to tell me what type of cell it is, okay, and what pathway it's going towards, what pathway it's sending its stuff through, and what that pathway does. Okay, here we go. So this one.
So it's an easy one to start with in the sense that it is in both sublamina A and B. So we know that's one of those K-type K-type ganglion cells, which is the coniocellular pathway.
Yep. And what does the coniocellular pathway deal with?
It's like proprioception
and which
blue yellow vision.
Okay. Here I was waiting. You're absolutely right. I said it like this, and you're absolutely right. It's the bistratified ganglion cell coniocellular pathway. Okay. But one thing I want to point out, these are the cells, because they're so new, they're actually very, very fragile. Okay. And so because they're fragile, there is one diagnosis that destroys the ganglion cell layer. What is the major diagnosis that destroys the ganglion cell layer? It starts with a G. An entire subspecialty that we have is on it.
Glaucoma.
Glaucoma, right? Okay. I just told you that's the ganglion cell layer. Okay. Well, who are they destroying? One of these cells. They're destroying these cells, right? Okay. I just told you this guy dies first. Okay, when you start compressing it, because he's got way more dendritic axons, more dies off. He's not as like hardcore, because he's got too many things that can go wrong. If you bust up one of these things, he's not going to be able to project well. So he tends to die faster. So whenever you have early glaucoma, what do they tell you to do? They tell you to do the blue yellow. Do you remember?
Oh, interesting. I'm not sure if I've gotten that tip yet, but I will now integrate it.
Now you know. Okay, so they'll often tell you to do that because it is an early, in their argumentation, an early sensing of these cells dying, and that tells you that things are happening, even if we can't necessarily measure the thinning of the ganglion cell layer. Make sense?
Yeah, totally.
So these are the preferential ones. Okay, so here's one. I'm going to go ahead and say that this is a really small kind of dendritic tree.
I was wondering how I was going to distinguish, but so it's in the sublamina, B and S345. And so this makes it an on cell. And if you're telling me it's very small, I'll call that a P-type midget cell.
Good. And what is the pathway called?
P for parvocellular pathway.
Parvocellular pathway deal with?
It is what's the largest set of ganglion cells. It's like color vision, slow conduction.
Got it. Red, green, we can specify here. Red, green vision, but you're right, color vision, red, green, and slow conduction. You got it, right? So I said that, and you said parvocellular pathway. It's going to the LGN. You just told me that. Good. Let's go and find this guy. I'm going to call him Giant.
Okay. Then if he's everywhere, then it's Giant. It's either is it the photo sensitive, this is the intrinsically photosensitive, retinal ganglion cell.
Yep. Or what is another word for them? What does it contain?
Melanopsin containing.
Melanopsin, right. And where would he project? Name one spot.
Anywhere but the LGN, so hypothalamus. Okay,
hypothalamus, yes. Anywhere but the LGN, but what we talked about. It's not projecting to I don't know, like one weird. But yes, you're absolutely right. Anywhere but the LGN. So like you said, hypothalamus, Edinger-Westphal, it deals with circadian rhythms, etc. suprachiasmatic nucleus. Okay, let's go here. I'm going to say this is a large, not a
Right.
large, it's not giant. It's like an
Right.
umbrella. Okay.
I think I'm picking up what you're throwing down. This
Right.
is so it's an M type, a parasol cell.
Yep.
It's in the, I guess that would be S1, S2, so it's off.
Yep.
And these project to the magnocellular pathway. They're like, I think you're saying it's large because it's like these parasol stands for umbrella. So this deals with like motion
Yep.
and fast responses. So it's like our rods.
Perfect. Yep. Fast conduction. You got rod. Exactly. And just remind me when you say off ganglion cell, what are you actually saying? It's shorthand for what?
It's off center on surround.
You got it. And even though we didn't explicitly go through that firing pattern, you could figure it out in your head through, not confusing yourself, but you kind of have the tools to do that.
Yep.
Okay. This guy I'm going to say is a large one again. I don't know why I did that. It's the same thing. Oh, I said the small. Okay, midget, whatever. Okay, okay, fine. That was a small one. Okay, you know it, right? Okay, so you got it. Okay, so that's that. Okay, so next time we're gonna legitimately go into the brain,
I
and
love it.
all those. Okay, I hope this is helpful. I'm gonna
This
send.
is awesome.
Yeah, and this is like I said. Now, just between you and me, I know that if you look at the OCT, you're going to be like, it's still confusing, but in your head, it's going to be like, okay, I know where the IPL is, and it's like, it's special, right? Like, it's not just some layer anymore, right? It's like, it has all these connections, right? It's okay. So I will let you know when we can do the next one, okay?
That sounds great. I will stay tuned. Thank you so much.
No problem. Okay, bye.
Stay easy. Bye.